I repeatedly have mentioned that computers are just humans trying to simulate life itself. Inside the cell, the organelle that is most like the CPU of a computer would be the nucleus. It has a housing, just like a computer tower or CPU. It has means for things to go in and out. It's got plugs where you can put electricity in and so forth. And that's where all of the programs are stored. That's where all the programs are processed. And everything comes in and out of cords or nuclear pores, in and out of the rest of the cell in order to do all of the processes themselves. The chromosomes are basically like this floppy disk. And this floppy disk comes from a time when we actually called them floppy disks because the disk inside was indeed floppy. Disk inside was indeed floppy. Floppy. I know I was always confused when somebody'd show me one of these and they'd say, this is a floppy, and it doesn't flop. Well, before that, the floppy disks actually did flop. And now one of the reasons that I have this stored in this sleeve is this is also an opportune time to talk about what are viruses. A virus only has two parts. A virus has the chromosomes, and they're stuck inside a protein coat, or it's also called a capsid. What this also helps do is this allows us to talk about how people develop immunity to colds or viruses. Basically, what we do when we've got when we're trying to do a vaccine for a virus is we take the virus, and remember a virus is a chromosome inside the protein coat. What we do is we take the chromosome and we somehow destroy it, either outside or inside, and we go ahead and leave it inside the protein coat. Then later, that virus, we take that destroyed chromosome virus and we inject it into the body. The white blood cells of our body identify the protein coat that's on the outside, and so anything that comes along that has the same protein coat, our white blood cells will attack and devour and eat to death. But anyhow, back to the inside the nucleus are little dark regions that we see whenever we look at it under the microscope. Those dark regions inside the nucleus are basically the same as the drives. The drives, and the purpose of the drives are to be able to read those chromosomes. So we can take or the discs. So we can take a piece of chromosome material, stick it inside the nucleus. It's read inside the nucleolus of the cell. The dark regions are the nucleoli, is plural. Nucleolus is one. And so the nucleoli have the purpose of reading the discs. Now, if we get more modern, this would be a replacement of what we would call a virus. It's got the coat on the outside, the protective barrier on the outside. Inside, it's got what would be the same as the chromosomes. Right now, this is what we call a DVD today, but it's basically the program. You can't sit at this and actually see this. This is not like a whole organism. This is just something that has the code, like DNA is code. It just has the code for being able to see this movie that was called distillation. If we do something to it like destroy it, a microwave will do a nice job of destroying it. Now we have a computer program that is totally destroyed. The microwave has totally destroyed that. You'd never be able to read that. We stick that back inside the protein coat, and now it's still inside the protein coat, but it can't do any of its functions when put inside our body or inside a cell. And so now our white blood cells can find this protein coat, identify it, attack and kill it instead of our own cells. And if we see any new healthy viruses in our body, they're already trained for this particular protein coat and we'll be able to devour it on sight. Sometimes I've heard people call water balloons or cells a lot like water balloons, and I don't think that's altogether accurate. There are a couple of reasons that I first of all am not comfortable with calling a cell a lot like a water balloon. First of all, the cytoplasm inside the cell is not a liquid and it's not a solution. Basically, we know there are three kinds of mixes. There is suspension, solution, and the third kind of mix is called a colloid. And a solution is like our soda pops and so forth that never settle out. A suspension is like that hot cocoa you had this morning that you had to get a stir stick because it would not stay stirred. And a colloid is kind of like jello, where things are like suspended in a matrix-like structure. So cytoplasm we know to be a colloid. It is not a solution, it is not a suspension, it's a colloid. The blue water balloon is a normal water balloon. The blue water balloon is a colloid-filled water balloon. The red balloon is a regular water balloon. And I think you might be able to see that if I bounce the red one, see it's a lot more jiggly, it's a lot different substance, it's a lot longer before it gives up the elasticity effect. You take the colloid balloon and you see it's completely different structure. Of course, this one is a little bit weird. If you make one of these, pass them around, teenage males start doing weird things with them. They don't want to give them up. But that's a colloid. And that's a solution. So this is your typical water balloon. So you can see that because cytoplasm is a colloid, that it's not altogether similar. One of the other concerns I have with using a water balloon as the same as a cell, or considering it the same as the cell, is that a cell has holes in it. It has micromolecule passing holes called pinocytotic vesicles. Pinocytotic vesicles have to let things in and out of a cell. A cell has to be able to eat through those holes. A water balloon does not have any holes in it. It is not able to take things in it. Now, if I take this water balloon, that does remind me of the old joke about I'll bet you $20 I can poke this needle in this water balloon and it won't pop. And of course, once they've done the bet, then you turn it over and you show them that the water balloon does indeed have tape on it. And so when you stick a pin through that tape, the tape is a preventative to the tearing of the surface of the balloon, and you may make a nice fountain, but it doesn't pop. And so then you of course discard the balloon before it does have a chance to break loose of the tape and tear. On the other hand, if you do actually stick it, see now there's a hole. It's like a pinocytotic vesicle, except you can't put anything in through it. It can only let stuff out. So again, why the water balloon is not quite like a cell. Of course, if you do on the place where you don't have the tape, if you do stick the pen in it, of course it will indeed pop. As you just saw, or as you are about to see again, the typical water balloon, when it pops, looks like this. However, because the cytoplasm inside, a cytoplasm or a cell doesn't quite fall apart the same way. When it's filled with a colloid, like the cytoplasm is a colloid, when you pop it, its behavior will be completely different. One of my favorite ways of simulating centrioles is with these Chinese magic sticks. They look like this. They're considered Chinese magic sticks, and you can do certain things with them. But what I do is, first of all, I simulate the centrioles of the cell. Now, these are only going to be visible just like the chromosomes, and so these tassels here look like chromosomes on either side. You only see the chromosomes through the nucleus when the cell is about to divide. And true like that, the centrioles are only visible as the cell's about to divide. The centrioles are usually found paired together, adjacent to the nucleus, what's up in a cell, right? And so you will see the duplicated chromosomes and you will see the centrioles just on the exterior of the nucleus. Then the chromosomes will be joined together in their duplicates. The two duplicate pairs will be joined together. And then the centrioles, as the advancement of the duplication goes on, will spread to opposite sides of the cell while the chromosomes are still duplicated side by side. And then as the cell begins to divide, the two sets of chromosomes snap to the two centrioles in the two different locations. They pull apart, and then a membrane comes up in between, making two complete new identical cells. Just for fun are these sticks, and I take these sticks and so what they do is, see, I can take this one and pull it down, and if I don't like that one being down, I want this one down instead, I pull that one down and well, okay, now wait a minute. Okay, let's see, I can pull this one down, and then if I pull this, wait a minute, okay, I can pull this, but now if I pull this, let's see, okay, how about if I pull this and this, but then if I pull this, I don't know, because they're separate here, and I don't know, anyway, just for fun. If you'll notice, the peanut is altogether shaped very much like the mitochondria. When you look at the cell organelle chart, you'll notice that the peanut has an almost identical structure, and I think this is not by chance. A peanut is a very high energy critter. Mitochondria are they're the powerhouse of the cell. They're for creating energy inside the cell. And so that a peanut is full of energy, and that mitochondria are for processing energy and for the energetics of the cell. It's interesting, these mitochondria only come from our mother. Our mother's mitochondria are the only ones we have in our cells. The egg, as it travels down the oviduct, is carrying all of our mother's organelles. So if I take this peanut to demonstrate the energetics of a peanut and how energetic they can be, I can take a peanut and even a half a peanut or less than half a peanut. And by putting it on a definition of a calorie is the energy required to raise 1 liter of water 1 degree centigrade. So I take that peanut and I light that peanut on fire. If the wind will allow it. You'll notice that that peanut is burning quite intently. There are lots of oils available, and those oils are very high energy oils. And so the energetics of the peanut allow it to burn for a considerable period of time. And just like the mitochondria are for energy in our cell, so is a peanut for our nutrition. Some of the best oils nutritionally are peanuts, or any of the nuts actually. Almonds, cashews, all of the nut oils are particularly good. Now, if you ever got lost in a cave and your flashlight went dead and you still had some peanuts on hand, just stick something in your peanuts, light them on fire, and you'd be able to travel through the cave without any kind of a difficulty. But look at the length of time that this marginal piece of a peanut has been able to burn. Mitochondria, peanuts, I see them as one and the same, energy factories. The ribosomes of the body are considered, or the cell, are considered the factories. They are factories that make proteins. Remember, the definition of a protein is 2 or more amino acids joined together make a protein. And we have several different kinds of amino acids that we've discovered earlier. So if this represents a ribosome, this Bill is, we'll call him Richie Ribosome. Rich Richie, whatever. I see Rich Richie's pictures on the front. Benjamin Richie. Okay. Richie Ribosome. And so Benjamin Ribosome, there we go, Benjamin Ribosome. Benjamin Ribosome it takes 1 amino acid. If we take him and fold him in 1/3, it takes 1 amino acid, and then he takes a second amino acid as it travels along. Let's say this is amino acid orange, or valine, or histidine, and then this one maybe is serine, or blue. It takes amino acid blue and amino acid orange, and the job is to take those 2 amino acids and join them into 1 molecule, and that 1 molecule with the 2 pieces joined together we call a protein, because it's 2 or more amino acids joined together. And the job of Richie or Benjamin Ribosome is to take 2 or more amino acids and join them together into strings by the code of the RNA coming from the DNA. The DNA is inside the nucleus. The RNA leaves the nucleus and goes out into the cell, meets Richie ribosomes on the edge of the endoplasmic reticulum, and Richies make the rough look on the endoplasmic reticulum. And so now Benjamin Ribosome, or Richie Ribosome, manufactures on the rough spots on the endoplasmic reticulum proteins. Of course, if you remember my dilemma about the cell membrane having holes, I think that a handy wipe is probably one of the best simulations I can think of for a cell membrane. You can see a handy wipe has the porosity. You can stick this paper clip in and out of it. So those holes in there might be the same as pinocytotic vesicles, and yet the cell's able to maintain itself without leaking all of its contents out. I've already used this jar as a partial example of a cell. Good. And of course we all know the trick where you take the non-soluble card, you put it over the top, hold it upside down, and because The surface tension around the edge creates a decent seal, and because the equal pressure outside is inside, actually it's a lesser pressure in the air in there because water is a hydraulic fluid. You see that the air outside is actually pushing this against that, and so it doesn't leak out. But we don't have to have something that has no holes in it. We could easily do that with our simulated cell membrane or our handy wipe. We take this handy wipe with all of its holes in it. You see it's got holes all over it. We can take that paper clip and stab it through all those holes. Yet if we take it with all those holes, when we turn it upside down, you're going to see that because of surface tension and because the size of the holes are not too big, you can see that because of the surface tension and the holes are penocytotic vesicles or no, the water is actually staying in there. And of course, since it does have holes, then that makes it a possible material that we could, well, the food of every cell is of course glucose, and so we can take that sugar or glucose, and in the presence of water, which allows things to go through membranes, in the presence of water, we can add water to it, and the sugar is allowed to transport directly through all those penocytotic vesicles and actually becomes inside with a little insulin too inside. So the cell membrane actually has pores that allow air and nutrients to go in and out of the cell. Basically, this is the nutrition of every cell that lives. It's called sugar or glucose, C6H12O6. Does that ever make you wonder about the symbol on C and H sugar? Notice that there's a circle around it for the O, there's an H, and there's a C. And what is sugar but C6H12O6? The people that named C and H sugar must have known the molecules of its contents. This is nothing other than an edible red vine. You know, that red licorice. It is a good analogy for endoplasmic reticulum. It has folds all over them. Endoplasmic means in the cytoplasm, or endoplasmic is in the cytoplasm, endoplasmic. And reticulum means folds or inundations. And of course, if you look at the surface of this red licorice or red vine, you will notice it has lots of folds and undulations. So for the reticulum part, it's well fitted, and it is a tube or a channel for transport. Endoplasmic reticulum are the transport channels. Now, a name like endoplasmic reticulum, what length and excessiveness it is. You know, scientists don't like that any more than you do. So the reticulum of endoplasmic reticulum is too much for most scientists. They don't run around going, oh, the endoplasmic reticulum was fine. Endoplasmic reticulum. They don't do that. So they call it ER. They do an acronym just like everybody else. They call it ER. And so another acronym I know of that's kind of fun is, when my students recognize it as ER, they're talking about edible red vines. ER. Edible red vines. It would be ridiculous to keep calling them edible red vines. Can I have some edible red vines? You wouldn't say edible red vines. You call them ER. So the analogy is good there. We'll show you another aspect of the analogy. There are two more organelles. Actually, one is a subset of the other that I would like to talk about. One is called a vacuole. A vacuole is nothing more than a storage chamber, just like this empty 2-liter bottle. If I wanted to, I could exhale things into the 2-liter bottle. And so gases that I didn't want inside the cell, I could put inside there. I could take some edible red vine. I could put some edible red vine in there and have food inside. As far as that goes, I could take and I could, well, do something enough and fill it inside with excretion and export that to the outside of the cell. I could even carry liquid in it. I can carry water in it. I could take and pour water inside it and be able to store water with it. So this is a vacuole. This is a very elemental part of many cells. It's called a vacuole. As I mentioned, I said that one of the organelles I talk about is a subset of another one. One of the very specialized kinds of vacuoles is a kind of vacuole called a lysosome. A lysosome contains a very special liquid that digests and breaks up cell organelles if the cell should die, so that other cells can use the parts. It takes like the ER, or edible red vines, or the endoplasmic reticulum, and it breaks them into pieces. Then it digests those broken pieces so that the rest of the cell can use them. If I were to take some lysozyme and pour it into a container, if you're ever doing this and you have a problem, you can throw it in that container. And of course, what do you think is going to happen to the ER that remains in that container? Of course, the lysosome. Lysis is a phrase that many of us may not know it at the outcome. Lysis means to cut or to digest or dissolve. And so one of the best things that I can think of is there was a soft drink called Slice. And so basically slice means to lyse. Slice, lyse. Slice is the same as to lyse. And so what will happen during the duration of that is that ER will be digested or sliced or lysed by the lysozyme that was inside the lysosome. The lysosome had lysozyme, which digests things. As we wait for our ER to be digested by the lysozyme, I do have yet one more opportunity to talk about the purpose and benefit of ER, or the functionality of ER, edible red vines endoplasmic reticulum. There's something that's been characteristic throughout time in the United States. Edible red vines have been with us for probably 50, 60 years. They are a hollow, reticulated structure, and many people, usually of cherry flavor or strawberry flavor. There have been people over the years that have used soda pops and the ER to flavor their drinks and as a straw, and that is particularly analogous to the purpose of endoplasmic reticulum. Endoplasmic reticulum are transport tubes. They're the freeways, they're the highways, they're the byways of the cell. And so if I were to take this edible red vine, or ER, and drink through it, I'm transporting water. That's nice tasting water. I'm transporting the water from something like a lysosome into my stomach. So that later I will be able to further digest the Even along the way, you'll notice it's digesting the ER. Now I have enough liquid to digest the ER. Once upon a time, I was a pharmaceutical sales representative. I was trained that if you do not say your product name at least 7 times in front of that doctor, he will not only won't he be able to write the prescription for it, he won't be able to even remember it. So you had to say 7 to 9 times the name of your product in the sales presentation, or it was a worthless presentation. Many people are not aware of the Golgi complex, or what the Golgi complex is. If you were to walk up to a person and said, "Do you have any idea what you do to my Golgi complex?" If they knew what you were saying, they would definitely slap you in the face. It's sort of like saying, "You make me wet," or "You make me juicy." The Golgi complex are special organelles. This is an analogous Golgi complex. They have a cell membrane folded inside the cell, and they are the secretory parts of any cell. They are organelles that release saliva in the salivary glands, or mucus in the sinuses, or the intestinal villi. And so what I'm going to do is I'm going to try to help you be able to remember the name, because remembering the name is as much as anything. I'm going to do the function of Golgi complex attached to its name 7 to 9 times. So basically they have liquid inside them, or some sort of excretory substance. And so in your anus, when the lubricants are involved, the Golgi complex are releasing the lubricants. And inside your intestinal villi, the digestive enzymes are released by your Golgi complex. When you have a runny nose, the Golgi complex is releasing additional mucus juices. When you're about to cry, in your tear ducts, the Golgi complexes are releasing nice high saline washing solution for your eyes. The Golgi complexes are very good at cleansing your eyes and cleansing your nose. The Golgi complexes in your salivary glands are releasing saliva just at the thought of edible red vines, endoplasmic reticulum, IER. Just think about a dog and the saliva and the juices exuding from his Golgi complexes all over his tongue, all throughout his mouth. Even in your ears, the earwax is released by Golgi complexes in the cells that line your ears. Golgi complex, Golgi complex, Golgi complex. Notice that as that piece of ER has resided in the lysozyme, it has begun to be digested by the lysozyme, which is just exactly what the lysozyme inside the lysosome is intended to do. Goodbye, ER.